All right. All right. Good evening and welcome to this live WebEx that would just check in to see if there's questions and things that we could address here in person, and also a bit of a review for our next exam that we'll let's see, we'll open up tomorrow, probably midday, and be open through Monday. So there's a decent window there for you to find a good time to focus, avoid distractions, have a good connection, have a decent lit room, and focus on just taking the exam. Just want to remind everybody, it is important to follow those steps with the Respondus Lockdown browser. As faculty, we are committed to ensuring academic integrity of everyone at the college, and it's my responsibility, your responsibility to do that, to do what needs to be done. And it takes a little bit of getting used to, but you should had two, at least for this class, two times with the lockdown, the practice quiz, and then the last exam. And a few people I needed to write follow-up emails saying, well, you didn't follow directions, please do it next time. You didn't show your NID to make sure that you are the person that's in the course taking the exam, and to stay in view the whole exam, and doing the quick kind of environmental check to show that you don't have notes and books and other people around to assist, right? It's your education, it's your uh, uh, careers, and it's your conscience to do things right. Okay. Um, before we get started going through I have some of our outlines that for the material to look at and talk through, see if there's questions, comments on, maybe just in while I'm reviewing the outlines, maybe something will come to mind, or some additional connections will be made if you hear me say it again. Maybe dispel some misconceptions, or you see another connection or so. Is there now, I'll stay on after we go through things if you have a specific question you want to ask me individually, that's fine too. Any general questions that might be helpful to all to ask and for me to try to answer? Okay. So I don't have the chat window open, so please, if you have something that that you want to say, please just just put your speaker on and voice, or give me a signal that you have something to ask or say. So this second exam goes into a little more in depth of the three macronutrients: carbohydrates, lipids, and proteins. Mind-tap, those exercises are important to review, help you reread parts of the text, introduce some real applications of what you're learning that hopefully will keep you curious about the topic of nutrition. Sometimes help you relate to something that you experience, and keep us interested, because it is a critical topic for us to continually be thinking about. Okay, mind tap, lockdown browser. Okay. These are pretty detailed outlines that are in the additional resources, and I thought that um, you have recordings that I've gone through, PowerPoints. You have the PDFs of the PowerPoints. So this is in a condensed version of all those topics. And these are very good outlines to review after you've reviewed the figures and tables and done the exercises, to now look at these terms, these sentences, phrases, uh, and see what comes to mind. Maybe something that you didn't really catch, but these are important points, not a tremendous amount of, but a lot in these little points and then the sub points below them. So I have the main portions for the three macronutrients, the outlines right here to talk through a little bit. Then I have um, the um, the study it little outlines, Whitney chapters, study it. That's a little more colorful than this black and white, but that also pulls out high points from the chapters and asks some questions, some multiple choice questions, and some little discussion type things to think about. And so it's good to go through the details, whatever practice you do, take notes or electronically or in a notebook, do diagrams and things. Yeah, there could be, it could feel like there's a lot of content, but it's presented in a way that we kind of do a spiral, hitting upon the same kind of themes a few times through the course. The chapter 3 in particular was it included um, representatives of most of the classes of nutrients, and specifically about the digestion, absorption, transport of them. Just a little bit kind of in general to show big picture, talked about the GI tract, and begin to introduce hormones, some signaling that's important to realize that things go on. Now, of course, you should realize that we're hitting on, it sounds like a lot of detail that we discuss, um, but as is always the case with a introductory kind of broad kind of course, we're touching upon uh, the top most important to get a general good kind of picture of things. Below the surface, there's much more that goes on. If you go into a field, as some of you have said, um, nursing, uh, dental hygiene, or dental technician, or any related health profession, um, these are very important concepts to have. And then, depending on your specialty, you might dig a little deeper in topics in nutrition that still align with your main career or profession or specialty. Okay, so carbohydrates. and the three main carbohydrates, sugars, starches, and fibers. So when we say sugars, that's kind of almost like a very general term, but that includes uh, table sugar, that's sucrose. It includes the other disaccharides that are in common foods like milk sugar, lactose, malt sugar. It's in malt and in beer and some other types of beverages. So these disaccharides, of course, they're made up of two of the monosaccharides. Very rarely does food have strictly monosaccharides. It's usually as disaccharides or longer polymers. That is a number of the monosaccharides linked with a saccharide bond of type. The monosaccharides have glucose, fructose, and galactose. Those are the ones we commonly know associated with foods, right? Glucose, of course, the importance of glucose is that all carbohydrates, to be useful for us, they're converted to glucose, even if it's fructose corn syrup. We do have to manipulate it and change it and eventually get it to glucose. And glucose is the molecule that goes in and goes through cycles glycolysis and TCA to eventually give us our energy currency, ATP. And that's coming up in chapters 6 and 7, our energy systems. So we mentioned monosaccharides, disaccharides, and the polysaccharides. That's just anything of a longer chain, which might include, and commonly in foods, starches. So starches, very long chains of repeating glucose units. And that's in common starchy foods. Can you explain the difference? Yeah, I didn't catch the whole thing. I'm looking for the chat. The difference between.
That was me. I asked the difference between glucose and glycogen.
Okay, good. Okay, glucose is the monosaccharide that we mentioned that we eventually break down disaccharides, starches that we get in foods. We break them into glucose units, monosaccharides, and then we store the glucose. The way we store it is we store it as glycogen. Glycogen is repeating units again of glucose, and that's mainly in the liver and also in certain muscle. Yeah. So glycogen is the animal, if you might say, or certainly human and other species ways to store carbohydrates, whereas plants store it as starches, and that they're similar, although starches have slightly different structures in the way they're more straight-chained, where glycogen that we store are more branched. And the theory is they're more branched because they're more hydrated. There's water molecules that are situated around the chains of glucose, as opposed to in plants, the starches, the chains are relatively straight with some branching. Okay, and we have specialized enzymes that are activated when our body needs more glucose put into the bloodstream. and that's signaled by the hormone glucagon. Hormone glucagon signals that we need more glucose put out into the bloodstream for tissues and everything everywhere else. The opposing enzyme of glycogen of glucagon is insulin. So insulin's put out when we need to get the glucose back into tissue or even back to the liver. And we maintain that pretty narrow concentration of glucose in the bloodstream by mainly these two hormones, um, uh, with some other kinds of messengers that are involved too that get much more involved. Yeah, Sully.
I know that insulin's secreted by the pancreas, but is glucagon also secreted by the pancreas?
Yes, it is. It's not
Okay.
exactly the same cells, but they're in the same area. The aisles of what are we calling that? I forget. Oh, so slightly different parts of the pancreas. Yes, generate these. Pancreas is extremely important, obviously. So that's why pancreatic disease or cancers are so complicated and devastating. When I had a friend in college who he's a great athlete. I used to play basketball with him all the time. He was ahead of me. He was like a mentor for me in graduate school. And then he got cancer, and it was pancreatic cancer. And it was such a short time that he was just overtaken by it. I think it was like less than a year, like a half a year, and he passed away from pancreatic cancer. So uh, it's difficult. Uh, uh, okay, following a little more here, major types of fiber. Okay, fiber is that's a general term. There's different kinds of fiber, of course, but this is generally that we cannot break the fiber down by our own enzymes. So it serves a very important purpose of helping the GI tract keeping it having bulk and keeping it moving. It's one of the nutrients that majority of people don't meet the recommended amount of fiber. It's interesting to look at our compilation and see, or even think about during the day, where are we getting our fiber? What types of food have this mostly indigestible carbohydrate that's important for us? The other important, and scientists are learning much more about how important fiber is the food for our microbiome. And that's I was just speaking with some graduate students at Princeton today via Zoom. We're getting them to speak at our high school in a few weeks, and there's a group uh, at in Princeton chemistry doing cutting edge research, as many labs are, about the microbiome and what exactly are the best kinds of probiotics, that is, and even prebiotics. But what's the food for this healthy microbiome, and then what are the products that are given off by the microbiome that seem to be very healthful for us in terms of strong immune system, and some other health benefits. So that's the fiber. Um, the microbes can break down a lot of fiber. Some of it just gets put out as important bulk in our waste stream. So digestion and absorption, digestion of carbohydrate. We know that's the one that we had this question already. Where does maybe we didn't have it, but it might come up. What's the first part of digestion other than manually massacrating? And that would be in our mouth. We have sugars, simple sugars, start to be digested. They start to be digested into monosaccharides to some extent. There are reported some lipases also in salivary, but not as much as what we could start digesting in the mouth, some carbohydrate. But the bulk of carbohydrate digestion is makes it through the stomach, not digested, but in the small intestine, where pancreatic juices and along the walls of the small intestine have enzymes to start breaking down polysaccharides and disaccharides. Enzymes in general, they go into the name carbohydratases as a shorthand for enzymes. We use the substrate and then aces at the end, ASES at the end. No real need to, wow, got a little stink bug here. Anytime it gets warm, so these stink bugs come out and they find them crawling around. They're relatively harmless unless you try to squish them, and they stink. List of major uses of glucose. Okay, so important to keep that glucose concentration, because too much can be inflammatory, too little can be hypoglycemia is also dangerous. Could have low blood pressure. You could just faint and fall and hurt ourselves, that kind of thing. So that's pretty critical there. Major uses of glucose. Okay, the major use is translating. It gives us energy. It yes, as we said, it's stored in bulk as glycogen. Glucose also is could be used in other certain structures. Glycoproteins use as you can hear in it, glycogen. So there could be messenger molecules also. So they're important substrate. The major one is for energy. Hormones that maintain normal glucose, we mentioned those, glucagon and insulin. Types of diabetes. This is what's still, I guess, still called type 1 and type 2. And there's in the medical journals, medical rounds, they're starting to name a type 3 diabetes that is more involved. We'll probably hear about that more soon. But type 1 is mainly an inability to produce insulin. That means there's a defect of something not working in the pancreas. And the type 2, which is sometimes referred to as adult onset diabetes, that's where the same amount of insulin is not as effective in doing its job to let glucose into tissues. So that's this insensitivity, insulin insensitivity of tissues, or insulin tolerance. level. Glycemic load. So that's those are measurements of.
I have a quick question, sorry.
Oh yeah, sorry.
The when you were talking about the hormones used to maintain the normal blood glucose, are we going to see anything about epinephrine on the exam?
That's a good point. So that's a nervous system as opposed to a hormonal. It's good to know that as another hormone that's effective in uh, stimulating um, insulin insulin release. So that's a flight or fight kind of a hormone that also, yes, acts like a glucagon in stimulating release of glucose. Yeah, thanks for mentioning that.
Thank you.
Okay, what else? Glycemic loads, that's the impact of certain meals or foods on how much of glucose appears in the bloodstream. So a high glucose load would be maybe a meal of a lot of bread, a lot of simple sugars, candy, of course. The initially, at least, the gold standard for the highest um, the highest number. Now, this was done at interestingly in Australia. Australia has this lab that did the studies that put together lots and lots of tables of data on glycemic load of various foods, and at the top was white bread and some fruits that you wouldn't expect also at the top, like watermelon. But it depends on how much we're talking about. You might have um, a food that has a high glycemic uh, index, but in compared to the comparison of the whole meal, it might be minor. There's also influences of the mix of other constituents of a meal that tend to tamp down that glycemic effect. For instance, if we are at a restaurant and bread comes out first, it's of course not a great idea to eat a lot of bread or even eat bread. And then you might say, "Oh, I don't want butter or oil," but that's not a good idea because the butter or oil tends to slow down the glycemic um, uh, effect. Okay, so it's a good idea to dip it in the olive oil if you're having bread or having a little butter on the bread before the meal. It's not as healthy just to eat the straight bread, thinking that you're not going to have the extra fat, a little bit of fat. Um, other aspects of carbohydrates here. Um, on dental health, of course, excess carbohydrate. Well, that's kind of food for microbes on the teeth and could put out, as they metabolize it, they leave some acid, which, if not rinsed off or washed off, starts to create harm in our tooth enamel and and so dental health related to sugar as well. What about alternatives to sugar? So there's a number of them, of course, and there's debate on both sides of if it helps saving the sugar calories or the sugar load, and having some of the uh, sugar replacements, the non-absorbable sugars or aspartame or those kind of things. Um, uh, well, the calories from a little bit of sugar that's put into whatever tea or soda calories is not the thing. It's the spike of sucrose that is the main culprit. The sugar that comes along with a burst of glucose into the bloodstream, not the calories necessarily. So that would speak to the alternatives. Might be a good option if we need that sweetness in what we're drinking. There's recommendations certainly for this DRI for generally for carbohydrate. There's some number, I don't have to remember an absolute number, but it's something like 300 or 350 milligrams, grams of per day if we're carbo counting. But it's important to have these, as we mentioned here, the fiber very important. The number is about 25 to 30 grams of fiber every day. And if you look at the components of your food, it's for lots of people, it doesn't add up to that much. So we should make a conscious effort to get that kind of fiber in our diet. It's in addition to the benefits of keeping the GI tract moving. It might be cleansing the GI tract. It's food for microbiome, but it's also having kind of in some instances a fulling effect or a satiating effect with the food. This is from that other outline resource that you have in additional resources, and this is this study put out by the textbook, but no longer in the newer edition of the textbook. So there's copies of this in additional resources. Good to go through these kinds of questions and outline what happens in a condensation and a hydrolysis reaction. So these are a few of the chemistry terms we come across in our course. Condensation versus hydrolysis. So if you look at hydrolysis, that's hydro or water. Lysis means to break. So it's using water to break a bond, hydrolysis. So as in our enzymes catalyze the hydrolysis of starch, for instance. So it breaks the starch into smaller units. That's hydrolysis. The opposite is condensation. Condensation reaction is it's the opposite. It's putting together. It's kind of making bonds so that it releases water and makes a bond. So we could see these in the formation of glucagon, of glycogen, when when our glucose forms glycogen. That's a condensation reaction. We see condensation reactions when we reform triglycerides in the liver. We store fat as triglycerides, but we break them down to absorb the fatty acids and the backbone, the glycerol backbone to absorb. So we're doing these hydrolysis and condensation reactions all the time with our macronutrients. Okay, we said something about starch and glycogen and how they're similar. They're similar in that they're polymers of glucose, and starch is how plants do it, and glycogen is how we do it. Okay, and they're both similar in that they're storage of glucose. Glucose, the substrate that we use for energy production. As I said, that's chapter 6 and 7. We talk a lot about that. Disaccharides, disaccharides include which? This is a question. So starch is a polymer, much longer. Glycogen is a polymer. Those are the long ones. Amylose, pectin, and dextrose. Let's look at the other ones here. We're looking for disaccharides. Sucrose, maltose, lactose. So that's the one I mentioned. These are the common disaccharides in our foods. Glucose, galactose, and fructose. That's the monosaccharides. Okay. Amylose, pectin, these are polysaccharides for the most part. Dextrose is another term for sucrose, table sugar. Pectin is a carbohydrate. You might know if you make jams and jellies, use pectin to kind of spread out make gels in your jams and jellies. So pectin, to get that from fruits like apples. Let's see what's here. Making a disaccharide from two monosaccharides. Okay, we just mentioned that. That's a chemistry kind of question there. And that's this condensation. What else do we have here? Digestion, hydrolysis, gluconeogenesis. Wow, that's a cool word. Let's see. That's we talk a lot about that in the energy chapter, but I think it may be mentioned. So, genesis, neogenesis, that sounds like we make something. We're making glucose. So, making glucose from non-carbohydrate sources. So, this is making glucose when we're starved of glucose. We might run out of glycogen, run out of glucose in our bloodstream. Then we have mechanisms to make glucose. Why? We need a certain minimum of glucose all the time because it's understood that our brain mainly only runs on glucose. It needs a minimum amount to perform its functions, and also certain tissues like red blood cells and some nervous systems are said to need this constant, at least minimum amount of glucose to function properly. So gluconeogenesis, we take something like some portion. We could take glycerol, the backbone of fatty acids, to make glucose. We can also make glucose from certain amino acids. So there's not a whole lot of things we could use to make glucose, but we have these backup mechanisms to do so. It's not a great thing for our system to use protein, to break up protein into amino acid and then use it to make carbohydrate. It's because it's breaking down protein. All of the protein, as we understand it, all the amino acids and protein have a function in our body. It's doing something. It's not storage. So when we start using protein, we're breaking something down. First, it might be muscle, then it might be other protein, and that's not good to go on too long in that doing that kind of thing. Starch and cellulose. So starch, of course, we can break down. We have the enzymes, whereas cellulose we don't break down. That's part of fiber. Okay. Glucose in the body. So we mentioned that. Here's a question. Storage form of glucose is a bunch of words here. And so these two trying to trick you here, glucagon and glycogen. They look a little bit similar, but we store it as glycogen. Maltose is that disaccharide found in malts or mostly beer. Insulin hormone that signals to glucose to be absorbed into tissues. Yeah. Insufficient glucose and metabolism, fat fragments combine to form. Okay, so this is in the case of um, low carbohydrate intake or use of all the glycogen. For instance, if you're running a marathon, you use up all the stored glucose. Then we do this. We form these ketone bodies. Ketone bodies that could replace glucose for a short amount of time as energy for the brain. Okay, ketone bodies can get through to the brain and be used for a short amount of time as energy in the absence of glucose. So that's the importance there. Yeah, that's an aspect of ketone-based diet or regimens to create ketone bodies. It does burn up fat, but there's a downside to that in that while that happens, there's some acid or pH goes lower in the bloodstream, and there's side effects such as nausea and some other things. And so, yeah. Pancreas secrete when glucose rises, when glucose falls. Okay, so that in that case, it's insulin when glucose is high, and when glucose falls, it's glucagon. So those are the main. And here's epinephrine is mentioned here, as Devin mentioned earlier. Epinephrine also similar to glucagon in its effect. So a little bit more here on digestion and absorption of carbohydrates. We mentioned a bit about the role of fiber in digestion and absorption. So that's a good question, be able to discuss about a little. Lactose intolerance. Okay, so lactose, as most of us know, is the common sugar or disaccharide in milk, and a large part of the population of the world is as you age, you might lose the ability to effectively cleave or to break the lactose into its galactose and glucose, into its monosaccharides. And the problem comes in when this disaccharide goes down to the microbes in our lower GI tract. The lactose gets metabolized, and the microbes form gas when they're digesting the lactose. Lactose also present in the lower GI tract could bring in more water into the GI tract. So that's why those two symptoms might affect people with some level of lactose intolerance. That means gas and diarrhea could be symptoms there. Ultimate goal of carbohydrate digestion absorption is to yield, and that answer is glucose. Amylase is an enzyme that I did not remember earlier. That's in our mouth. A real quick digestion, or it's an enzyme for simple sugars that digest them in the mouth, and we could start absorbing things very quickly. Even in the mouth, it's said that some glucose monosaccharides could be absorbed into our bloodstream, even in the mouth. Enzyme that breaks disaccharides into glucose and galactose. Okay, glucose and galactose. So that is maltose, maltase. That's kind of a detail that you don't have to be memorizing necessarily the makeup of each disaccharide, just knowing that they're disaccharide and they're made up of those three monosaccharides. Lactose, glucose, and fructose. Okay, did I say that wrong? And types of foods that provide starches and fibers. So starchy vegetables or grain. Fibers would be in whole grain would have more of the fibers. So in processing of wheats and flours, some of these whole grains are taken out that contribute to better fiber. So usually the lighter bread is less healthy in this regard, and more strictly very quickly digestible, higher glycemic load kind of carbohydrate. Okay, found in all foods except carbohydrates. So when you look at, that's why I had that picture of a plate when we first started. When you're looking at your plate, I wonder, do I know where the carbohydrates, where the fats are, and where the protein is, and where it's balanced. And the colored stuff usually indicates that we have possibility of having vitamins and well, vitamins mostly. Let's see, percentage of daily intake should come from carbohydrate. The carbohydrate is the one that we suggested that you have the highest percentage of calories. So that answer is D here. Okay, triglycerides. So I think, well, they're all varied, all the macromolecules, macronutrients rather. So to know at least these points on this guide is a good way to make sure that you have learned this content. So triglycerides are the major form of lipids, which also could be exchanged for fats. Lipids is a little more technical term. Phospholipids, they're in foods, but they're more rare in foods. Phospholipids, so that has a phosphoric acid end, and on the fatty acids, on some of the fatty acids, but it's also still a triglyceride. And sterols, these are kind of closed rings, higher molecular weight, it's still considered fats. They're hydrocarbons, and they're from plants. So plants, triglycerides mainly from animal food sources. Phospholipids could be both. Yes, there could be triglycerides, minor amounts in things other than animals food. Triglycerides consist of that 3-carbon backbone called glycerol, and then there's 3 fatty acids. Fatty acids are of different carbon number lengths. It could be typically from 8 carbons long up to 16 or 18 carbon long. So there's three of those, three fatty acids, and they could vary in the number of double bonds. So most of them have single bonds, and when they have a double bond, we call that a point of unsaturation. Saturation means that it's saturated with hydrogens. So each carbon connected to two other carbons, then it has the other two bonds are hydrogens. So just review your picture of that. Don't have a picture right here. But it's saturated. The term saturated implies we're saturating it with hydrogen. The double bonds are seen to be healthier type lipids when they have at least mono or poly unsaturated. Polyunsaturated. And so saturated fats are more solid. Unsaturated fats are more of the liquid type. So main fats that we get from animal food sources are mainly saturated fats. That is, they're more solid. And plant sources are more liquid. There's more unsaturation. Some health benefits are seen, and these are even essential fatty acids, omega-3s, omega-6 fatty acids found in fish oil or fish, and some plant sources like flaxseed, and some other sources could be nuts, and mainly some fatty fish. The omega-3, omega-6 refers to from the end of a fatty acid, from the end carbon, numbering that 1, and then counting 1, 2, 3, and a double bond. That would be omega-3. If we count to 6, that would be where the double bond is. That's the omega-6. Combinations of these are seen to be healthy to have in our diet regularly. And there's debate over the ratio. What's the perfect ratio of omega-3 to omega-6? It's not really totally agreed on, but it's known that these are essential for various health benefits, protection against heart disease in the long run. Let's see. Okay, so digestion, absorption, transport of lipids. They're of course to be absorbed. They go a different route. Rather than into the bloodstream, they go into the. Oops. They're broken into the fatty acids and glycerol instead of the bloodstream. They are into that other transport system. What is that? Right now.
Is that the lymph?
Yes, lymph, right, good. The lymphatic system. And in the pictures that you see in the book there, the red and blue are the bloodstream, and the lymph is pictured as yellow. And the lymph, the difference, there was a question or two in the last test from chapter 3, the lymphatic system is not by the pump the way the circulatory is, but it's a one-way kind of, when material is in it, it moves, slowly moves until it's in a form that could be put out into the bloodstream. And those are those different forms of lipoproteins. The largest is called the chylomicrons. The chylomicrons is a big way to move fatty acids and cholesterol and some protein, and then mainly to the liver, and the liver processes things and puts out from time to time these lipoproteins where cholesterol is needed or some fat is needed in various parts of the body. And those are the different kind of lipoproteins, the LDL, the HDL, and those HDL is that seen as the healthy lipoprotein that really picks up excess cholesterol and fatty acids and brings it back to the liver. So there's real dynamic moving around of free fatty acids and cholesterol. It's always these buses moving around the body. And if there's an overflow of certain types of fatty acids or too much cholesterol, that's where issues could arise of plaquing and sticking in arteries. That's where we hear the etiology of certain heart diseases. So there's fats. Fats are very important in the body. They're again building blocks of tissue. They're insulation. And of course they're used as energy also. It's kind of like a combustion engine has oil and gas. Well, you could think of it a bit like carbohydrate and fat. There's always kind of a mixture in our energy production. Okay, we mentioned about mono and polyunsaturated. Okay, so here's the other study guide that has some nice points, crisp points of reviewing things. Here's this a similar thing in the book. Here, the chylomicron is mainly triglyceride with some phospholipid, some cholesterol, and very little protein. And high density means more protein. So you see the high density lipoprotein has more protein, less cholesterol, more phospholipids, and these are the buses for fats and cholesterol in the body. Okay, mentioned most of this. Hydrogenation. That's a food processing technique. Hydrogenation that is done by food manufacturing to stabilize fats or to provide a certain type of texture they want in a food item. By hydrogenating across double bonds, you're taking away the unsaturated and making saturated fatty acids, thus changing more liquidy fats into stiffer or solid fats. In that process, sometimes trans fatty acids are produced, and those are unhealthy, found in animal models to cause possibility of cancers, radiation, and some other plaquing and such. So these are not good, and food manufacturers are required to state and analyze their processed foods for trans fatty acids and say what % of the fat is trans fatty acid. And it should be less than 1%. And they need to have that on a food label, any kind of processed fat containing food label. And hopefully everybody's more conscious, and if you didn't do before, to be curious and look at food nutrition facts labels. Let's see, digestion, absorption of lipids, and transport of lipids. We mentioned a bunch of that. So the digestion of fats, it's a little more complicated because they're not just hydrolyzed and put out into the bloodstream. Of course, they have to be carried, as we mentioned there, in the lymph nodes lymphatic system, but also in their digestion. They require bile to emulsify the fat. Why do we do that? Because we need to make it in a form so that the enzymes can get to the point of hydrolyzing the fatty acid from the glycerol. So the bile helps that out. Bile is formed in the liver and stored in the gallbladder. And when it's signaled, it's signaled by another digestive hormone that you learned about, and that's cholecystokinin, CCK, that says there's fat present and we need bile, and the CCK is the thing that gets things going, gets the bile present, and signals lipases and things for digestion. So a lot of coordination there that goes on. Okay, here's a nice review of the lipid family, all the members of the lipid family. Of course, there's health recommendations with regard to intakes of saturated fat seen as the less healthy fat. We definitely need certain amounts of fat. We know that there's the omega-3, omega-6s are required. We can't make those, and they're seen to be a necessary nutrient in our maintaining our health. Too much fat is not good. It's recommended that it's less than 10% of calories are in saturated fat. Trans fatty acids, trans fats, it's good to get none of those. There's seen to be certain health benefits of having regular amounts of mono and polyunsaturated fats, though it's not known exactly the amounts of each of these. So it's prudent for everybody to have sources of unsaturated fat and polyunsaturated fat and to know what they might be. It's interesting that monounsaturated fats, a lot of them, along with saturated, are in things like coconut oil. So things that you could think of that have some liquid and some solid fat. So coconut is a thing, but also the oils that we see as being healthy, olive oil, and even other canola oil seen, because they're unsaturated and polyunsaturated. Lecithin is an important phospholipid, really the only one that we typically mention by name. The others, unless you're a biochemist in the lipids fields, then you don't know them. Lecithin is one that is a phospholipid and important in membranes. Okay, membranes. Membranes, of course, could have a water-soluble side and not water-soluble side, and we know about the lipid bilayer around all cells. Very important to keep those remade as they are degraded. So keeping up all our systems, we need all those things available in pools if possible. So proteins are things, of course, they're made up of these units, amino acids, but amino acids are not things that just hang around in pools. They're temporary pools that are used and then they're degraded. So if we don't use the protein that we ingest and digest, then they're ultimately going to be destroyed and gotten rid of pretty relatively quickly, because they could become toxic. The amine groups could be reactive, and if they're not used to make protein, usable protein, then they're in effect denatured, but they're actually, the word is called deaminated. that is to get rid of the amine. To get rid of the amine, take that amine group and ultimately make it into urea. That's how we get rid of it. To combine with CO2. Okay, there's 9 essential amino acids, and we use 20 amino acids to make all the things on a regular basis, whether that's enzymes, whether that's muscle, whether that's bones, whether that's any other tissue that's damaged. We need these 20 amino acids. Nine of them, we can make the rest, those other 11. But we need those 20 on a regular basis to be available because we're constantly making various proteins. Because what? Because proteins are damaged and used and have to be replaced on a regular basis. Proteins, of course, I just mentioned a few of them there. It's of course structure, it's hormones, it's enzymes, it's part of bone, in fact, also. So amino acids, as it sounds, there's an amino side and there's an acid side, and then there's a group that differentiates them from one another, and that's sometimes called the side group or the R group that makes them different. Each of the 20 have different R groups. Here are the essential amino acids. You might recognize some of them, like tryptophan, that's mentioned often. Tryptophan, threonine, tryptophan, that's mentioned. That's supposedly a high amount as we digest turkey and gives us this sleepy effect. Okay. Absorption. So there's absorption of amino acids as we break them down. So protein digestion takes place in the stomach. You know, to the typical person that hasn't studied much about nutrition or physiology, might think everything is digested in the stomach. Of course, we know that's not right. It's mainly the protein that's digested in the stomach, and that's the HCl, hydrochloric acid, that's produced from the stomach wall, and that puts it in the right pH for denaturing enzymes to work. Because these proteases only work at low pH, like somewhere around pH 2. They come into the stomach not ready to do digestion until HCl has decreased the pH to 2. So it's a nicely working system. We don't want anything to be digested only when the conditions are right and we're sure that it's food that we're digesting and not ourselves. Okay, so we get enzymes from various places. It might pancreas, of course, delivers a bunch of enzymes, and even when the when it comes out of the stomach, the chyme is very acidic. It comes into the small intestine, the duodenum. Okay, it has a lot of protein that's digested, but it has all the other stuff that is not digested yet. The fat and some carbohydrate that still needs to continue to be digested. So pancreatic juices and bicarbonate comes into the small intestine and increases the pH back into the normal range or neutral range. So other enzymes could work on the other substrates like the fats and the carbohydrates. Okay. When the protein is in an amino acid form, then it has these particular places where it could be absorbed. Right. Proteins, there's a good list of the importance of protein in the body: structure, enzymes, hormones, fluid balance. The main constituent, one of the main constituents in the bloodstream, is protein, and that to keep fluid balance, keeps acid base. It acts as a buffer. Proteins are transport various things. Protein, of course, moves our essential gases around, the oxygen and CO2. That's in the form of that wonderful protein called hemoglobin. Hemoglobin, a more complex protein. That's the example that's usually used for a very complex protein because it has not only primary, secondary, tertiary, but quaternary structure. So it has 4 subunits around. So remember that. That's the hemoglobin. That represents among the more complex types of proteins. It has quaternary structure. And in those quaternary structures, each of the 4, it has iron in there, an iron atom. Okay. And for a moment there, I didn't remember the fluid balance in the bloods, and that's wow, I just forgot it again. The protein that's in bloodstream starts with A. Anybody remember that? Okay. Well, I wouldn't put it on the test. Okay. Protein can act as gluconeogenesis and could make glucose. It also is involved with blood clots, forming blood clots, that is, along with that special vitamin that almost doesn't sound like a vitamin K. It sounds like someone just made that up, but it is vitamin K involved in important in blood clots. Okay, these questions also on that review sheet there. Essential fatty acids, don't worry about naming the 9 of them, but there are 9 essential amino acids. You probably recognize some of them. The sequence. Okay, so the chapter goes into a little bit of how proteins are made, where it gets its instructions from from DNA, and how that then transcribes and turns out the primary sequence that eventually makes all protein. What do we have here? Isoleucine, leucine, and lysine are what? So these are among the essential amino acids. You might just recognize them. Oh, they're fatty acids. Amino acids. They're not proteases. That's enzymes. Polypeptides. So polypeptides are strings of amino acids. Short sequences of amino acids are called peptides. In fact, when our body condenses and makes a peptide bond, those are called peptides, when there's more than one amino acid connected to one another. Okay. So we talked about digestion in the stomach, protein in food. What's important about that? How do we evaluate how good a protein is in food? So there's kind of general parameters around quality of protein. It's got a few different things. It could be Digestibility, that's one kind of part of a quality protein. How easily can we digest it? And that varies among all of us. If you're a regular meat eater, then your system is kind of ready to digest meat. It almost expects it. So you have those proteases ready to go. But then other people that might not eat much meat or very infrequently are not ready to do that. I don't eat a whole lot of meat. I don't eat a lot of red meat. So when I do have occasional um, burger or something, then it takes me longer to digest it. I would never really eat steak anymore. That would take me too long to digest. I haven't eaten steak in many years. Okay, so quality protein can be gotten. It's not just meat. Egg is very seen as a high quality protein. Uh, corn is not. Corn has very little, if any, protein. Gelatin does have protein. It's not a complete protein. Whole grains are uh, not usually considered whole proteins. Whole protein means it has a full complement of essential amino acids, a whole quality protein. Okay, so we mentioned this already. In the stomach, hydrochloric acid does what? Um, so it it does not directly it denatures protein. It's not strong enough to actually break up the proteins. That's why we need the enzyme, and that's pepsin. It activates the pepsin. It was a pre-enzyme, and now it's ready to go. So it denatures the protein, gets it in a form that it can be more easily digested by the pepsin and some other proteases. How do we figure out how much protein do we need, and do we get enough protein? I think one of the exercises in MindTap asks you to think about this. There is a formula that generally applies, and it's uh, point eight grams per kilogram of body weight. So you'd have to know, translate if you're usually thinking in pounds to translate that into kilogram is 2.2 pounds. So that means if someone is about 150 pounds, let's say, and that's about 72 kilograms. So you multiply that by 0.8 grams per kilogram, and comes out to something around 60 to 65 or 70 grams of protein a day. Yes. So, question.
So would we have a question like that where we have to convert kilograms to pounds and then do pounds to milligrams or grams?
Yeah, no, you don't have to know unless I would give you the conversion factor. Um, is that So let's see what this question says. Protein RDA for healthy that weighs 180 grams, 180 pounds. Okay, so this one, yeah, this one would want you to make that conversion. So it's that one. 180 divided by 2.2 times Okay, that comes out to 65 grams of protein a day. I just did that calculation on that one. So I divided 180 by 2.2 kilograms per pound and multiplied by 0.8. So it's good to remember that number, 0.8 grams per kilogram. That's the requirement for us. All of us should have in mind. I mean, everybody is probably between 50 and 100 grams of protein a day. And if you look at your compilation of food and that kind of thing you eat during the day, hopefully if you're falling in somewhere around there, because a deficiency in protein, though it's mainly rare, it's not a good idea. In fact, there's some new studies coming out that uh, people that are on too much of a um, uh, plant-based without thinking of making sure they get protein is not good for the long term. You could just imagine why. If we don't have those amino acids present and we're not repairing muscle, we're not remaking enough bone, not remaking red blood cells in the long run, that could cause chronic conditions that we don't want. Okay, just a few more kind of visuals as we've been talking about the macronutrients. That's what the amino acid looks like right there. Whoop. Amino acid looks like right there. This is called an amine group, NH2. Here's the acid, and this is the side group, sometimes called an R group. That's what differentiates all amino acids from one another. Amino acids are unique in the units of that make up our macronutrients in that it contains nitrogen. There's guaranteed one of the questions on the test will be what's unique about amino acids or protein in terms of chemistry, and it's usually some kind of answer involving the nitrogen. Okay, because carbohydrates and lipids don't have nitrogen, so that's unique. And nitrogen is usually a more reactive atom, especially amines, and that's why we have to denature them and get rid of them, either use them or get rid of them. Okay, for all the macronutrients, really good study guide. This figure has lots of information in it for each of the macronutrients. So spend time to look at that and know all these terms and things that are on there. You don't necessarily have to memorize, but be able to recognize that trypsin and pepsin are talking about enzymes for digesting proteins. Again, we mentioned this already. Important functions of protein. Irreplaceable. Regarding quality, protein quality, does it have all the amino acids? Most of the time, foods like nuts together with whole grain and eggs have all those essential amino acids. That's no problem. You get them, especially with dairy, and with but some completely plant-based are missing some amino acids. And in the case of rice and beans, there's a reason why. I mean, historically, probably it comes from somewhere knowing that combining these two make it a quality meal. You could see that the beans, legumes, are missing methionine and tryptophan, okay? And the grains are missing isoleucine and lysine, but together, and I mean, like that, you get get all of them. And there's some examples like this of combinations of protein containing, but not necessarily complete protein, but put together, they will give us complete protein. I think I was saying whole protein before. It's complete. Complete meaning it has all essential amino acids in significant amounts. To compare, just as a structural comparison, the carbohydrates are made up mainly of carbon and hydrogen with some oxygen. Okay, I think that's what I wanted to go through. We went through study guides that you have that are really good to make a last kind of review of, including your notes. The questions, won't be exactly straightforward memorizing stuff, but applying things and answering some questions that might involve putting together some of these ideas. Something like by eating regularly, this kind of eating pattern, what might be missing? What's a potential nutritional deficiency? It might be that there's no fat there. You're not getting the essential fatty acids. It might be that there's very little protein. So it's putting together, a couple of the ideas that we're talking about. The test will be similar in that maybe 30, 35 multiple choice questions, and maybe 5 or 7 open-ended, kind of brief discussion type questions. Okay, so these are the macronutrients that we're studying here in this chapter. Any final questions or comments? Okay, so the protein that I was not remembering is albumin. Albumin is the main protein that controls the osmotic regulating fluids in blood. Albumin in blood. Okay. All right, so if no other comments and questions, thanks for your attention and being here. And yes, okay.
Thank you.
Okay, all right, you're welcome.
Thank you.
All right.
Have a nice night.